Why isn't there a universal CF gene therapy? Have you ever wondered why we don't have a universal gene therapy for cystic fibrosis? The answer lies in the complexity of the disease itself. Cystic fibrosis is caused by mutations in the cystic fibrosis transmembrane conductance regulator gene, commonly known as the CFTR gene. There are over 1,000 different mutations identified, and these mutations can affect the CFTR protein in various ways. Some mutations result in defective proteins, while others may produce no protein at all. This genetic diversity means that a single gene therapy would need to be effective across all these mutation types to be truly universal. Current treatments, such as CFTR modulator drugs, only work for patients whose mutations allow for some functional protein to be produced. Unfortunately, this leaves about 10 to 20% of patients with null mutations without effective treatments. Gene therapy aims to correct or replace the faulty CFTR gene at the DNA level, potentially offering a solution for all cystic fibrosis patients, regardless of their specific mutation. However, several significant challenges remain. One major hurdle is delivery efficiency. Getting gene editing tools or healthy CFTR genes into the affected lung cells is not straightforward. The lungs have protective barriers and immune defenses that limit the ability of viral or non-viral vectors to reach and enter the target cells. This makes it difficult to ensure that the therapy is delivered effectively. Another challenge is ensuring long-term expression and safety. For gene therapies to be effective, the corrected gene must be expressed consistently over the patient's lifetime. This needs to happen without causing harmful immune reactions or off-target effects. Achieving stable, long-lasting gene expression in lung tissue is still a major obstacle. Additionally, the body's immune system can react against the viral vectors or gene-editing machinery used in these therapies. This immune response can reduce the effectiveness of the treatment and raise safety concerns. The technical complexity and cost of developing these therapies also pose challenges. Creating effective gene therapies requires advanced technology and extensive research, making them expensive and limiting accessibility, especially in low- and middle-income countries. Despite nearly 30 years of research, clinical trials have yet to produce a fully successful gene therapy for cystic fibrosis lung disease. Researchers are continuously refining delivery methods, gene editing techniques like CRISPR, and vector design to overcome previous failures. On a positive note, ongoing research is promising. Advances in gene editing, personalized medicine, and stem cell therapy are paving the way for more effective treatments. Collaborative efforts among scientists, clinicians, and industry are crucial to overcoming these obstacles. In summary, the absence of a universal cystic fibrosis gene therapy reflects the complexity of cystic fibrosis genetics, biological barriers to effective gene delivery, safety considerations, and the technical and financial challenges of developing broadly applicable treatments. While progress is steady, more research and innovation are needed before a universal gene therapy can become a reality for everyone with cystic fibrosis. Always consult healthcare professionals for personalized medical advice and stay informed about the latest developments in research and clinical trials.